Okay, let's unpack this fundamental challenge in drug development. It's this constant tension, isn't it, between the urgent need for new medicines and the absolute imperative to make sure they're safe.
It absolutely is. That's the core dilemma.
Yeah, it really makes you think, how do we push progress faster, get those breakthroughs without, cutting corners on patient safety?
That's the tightrope walk. It's driven by huge unmet medical needs, the competitive landscape too, but safety has to be paramount every single step.
Exactly. And that's our deep dive today, this whole intricate balancing act innovation versus safety and bringing new drugs forward. We've got some great sources, preclinical handbooks, regulatory stuff like the CFR,
Initial rule books, yeah.
and even some really specific examples from organic process research. The real nitty-gritty. So the mission here is to pull out the key insights. How is this balance actually managed? We'll give you a clear picture, but, keep it engaging, avoid getting bogged down in jargon. Ready?
Absolutely. Let's start with those driving forces. You mentioned unmet needs. That's huge. Think about diseases with few or no options.
Right. The pressure is immense. And the competition between research groups, that fuels it too.
Definitely. But running right alongside that drive for speed is this non-negotiable commitment to safety. It's not just a box to tick for regulators.
No, it feels much more fundamental than that.
It is. It starts way before humans are involved. Preclinical investigations, the lab work, in vitro, and the animal studies, in vivo.
This is where you first look for trouble signs.
Exactly. Trying to figure out potential toxic effects. Which organs might be hit? How does the dose affect things? Is it reversible? It's so critical the FDA actually mandates it in 21 CFR 312.23A8. They need that non-clinical data to feel confident it's reasonably safe for that first human dose.
So that specific rule really underlines that you need solid preclinical safety data before you can even think about human trials.
Precisely. It's an early warning system, trying to predict, essentially.
And I saw in the Anti-Cancer Drug Development Guide, they use very specific tumor models in animals for efficacy tests. Things like pancreatic ductal adenocarcinomas, colon adenocarcinoma.
Yes, specific models, often chosen because they aren't easily rejected by the animal's immune system, or maybe they're resistant to current drugs. It helps get a clearer signal on whether the new drug actually has anti-cancer potential.
So it's about reliable indicators.
Right. Consistency. And importantly, all this preclinical data, especially the safety findings, directly feeds into setting that initial dose for human trials.
How does that work exactly?
Well, we look for something called the NOAEL, the No Observed Adverse Effect level, basically the highest dose in animal studies that didn't cause significant problems.
Oh.
Then you apply safety factors. You divide that NOAEL, often by quite a bit, to get a much lower cautious starting dose for humans.
So find the animal limit, then dial it way back for people. Sounds careful.
Yeah. Very. It has to be. Because you're introducing something completely new into a biological system. There's always inherent risk, uncertainty. Biology isn't always perfectly predictable.
No, definitely not.
Which is why development is so iterative. Findings from one stage really dictate how you design the next. If preclinical studies flag, say, a potential liver issue,
then the clinical trial protocol will specifically watch liver function like a hawk in the human participant.
Exactly. It's a step-by-step process, building evidence, always looking for issues.
And you mentioned predictive models earlier. It's not just about watching animals, right? There's more planning.
Oh, absolutely. Preclinical study design uses predictive models more and more to choose the best animal models, the right dose ranges, anticipate safety concerns. And in vitro models, lab-based tests are key for understanding ADME, absorption, distribution, metabolism, excretion. Basically, how the drug gets in, where it goes, how it's broken down, and how it gets out. Understanding that early, like we talked about with quality by design, is crucial for designing safer drugs.
Right. Which brings us neatly to the ethical side of things.
Yeah.
It's not just science and safety data, is it?
Not at all. There's a huge ethical dimension. Balancing that speed versus thoroughness. We can't rush things if risks aren't fully understood.
And in clinical trials, too.
Absolutely. How participants are recruited, informed consent, constant monitoring of their well-being. It's all governed by strict ethical principles. And as we said, using more in vitro models also helps ethically, potentially reducing animal testing.
It's a constant weighing of benefit versus risk, guided by ethics. Okay, so this whole complex process, it doesn't just happen in a vacuum. There are gatekeepers, right?
Major ones.
Yeah.
Regulatory bodies like the FDA here in the US or the EMA in Europe, they set the rules, enforce the standards for safety and efficacy. They're the critical checks and balances.
The ultimate decision makers, really. Really? They see all the data before anything gets approved?
They do. A huge step is submitting the IND, the Investigational New Drug Application, or the IMPD in Europe.
That's the big package of information.
It is. It compiles everything. Pharmacology, toxicology from those preclinical studies, chemistry details, manufacturing plans, quality control, and the detailed protocol for the first human trials. It's the foundation.
Wow. So you lay everything out before even the first dose in a human trial subject. That's intense vetting.
It needs to be. And even once trials start, the oversight continues. To get marketing approval, companies need substantial evidence, that's a legal term, from adequate and well-controlled studies. That's straight from regulations like 21 CFR 202.
Adequate and well-controlled. That means things like control groups, proper study design.
Exactly. Minimizing bias, getting reliable data on benefits and risks. And everything has to follow GCP, good clinical practice.
UGCP.
Yeah, international ethical and scientific quality standards. Think ICH E6, 21 CFR 314. It covers participant rights, safety, data integrity, the whole works. It ensures the trial results are credible. And it's similar for medical devices, too, under the CDRH branch of the FDA.
So pills, devices, same high bar for safety and effectiveness. Okay, let's make this more concrete. Can we look at specific examples, challenges where this innovation-safety balance really plays out?
Sure. Think about nanotechnology in drug formulations. It's innovative stuff, like using amorphous nanoparticles to help drugs dissolve better, which can boost absorption.
Sounds good. Better drug delivery.
It can be, but the challenge, the safety aspect, is stability. Are those tiny amorphous particles going to stay stable during manufacturing, on the shelf, or could they change back to a less soluble form that might reduce effectiveness, maybe even change the safety profile?
Ah, okay. So the innovation creates a new potential problem to solve.
Precisely. Researchers are looking at things like nanocrystals as potential solutions to keep things stable. Another big area is understanding ADME, what the body does to the drug.
Absorption, distribution, metabolism, excretion again.
Right. Things like lipophilicity, how fat-loving a drug is, affects how it moves through tissues. Or interactions with transport proteins like P-glycoprotein, these cellular gatekeepers. These factors massively influence safety and efficacy.
They're like tracking the drug's entire journey.
Exactly. Is it metabolized too fast? Or into something toxic? Does it interfere with other drugs? These are critical safety questions.
And even in making the drug itself, the chemistry. I saw mentions in the OPR&D literature about impurities.
Yes, that's a great example. One OPR&D article discussed making tosylamidoxam. During the process, a potentially harmful impurity, ethyl tosylate, could form.
Genotoxic, I think it said, meaning it could potentially damage DNA.
Correct. So while purification steps like crystallization remove most of it, the chemists have to be aware, understand how it forms, and develop ways to control it to incredibly low, safe levels. Sometimes they even change the whole process, like using citric acid as a catalyst to avoid making it in the first place.
So safety drives the actual chemistry design. It's not just about making the target molecule.
Absolutely. It's about the whole process and controlling anything potentially harmful.
What about really early on, finding drug candidates in the first place?
High-throughput screening, HTS, is key there. Testing thousands, millions of compounds quickly against a target.
Finding initial hits.
Right. Then medicinal chemists take those hits and start tweaking them. They want to improve activity, make the drug more selective for its target, but right away they're also thinking safety. Does it hit other targets unintentionally? That could cause side effects.
So precision is important from the start, not just hitting the target, but only the target, ideally.
Ideally, yes. And then there's formulation. Turning the drug substance into a usable medicine, a pill, an injection.
That has safety angles too.
Definitely. For inhaled drugs, getting a consistent dose to the lungs is crucial for efficacy and avoiding irritation. For injections, you need to adjust things like osmolarity, pH, so it's compatible with body fluids, doesn't cause pain or tissue damage. These details matter hugely for patient safety and tolerance.
It really drives home that safety isn't just one check. It's woven through everything, from molecule design to the final product. Now, you mentioned quality by design, QBD, earlier. How does that fit in?
QPD is a more systematic approach. It's about understanding and controlling product quality, including safety and efficacy attributes right from the beginning.
Building quality in, not just testing it at the end.
Exactly. You identify the critical quality attributes early, the things essential for safety and efficacy. You figure out what factors influence them. Then you design the drug and the manufacturing process to consistently hit those quality targets.
So it's more proactive, based on understanding the science and the process.
Yes, it allows you to anticipate and mitigate potential risks much better. It can lead to more robust products and potentially even smoother regulatory reviews because you understand your product so deeply.
Sounds like a smarter, safer way to develop drugs. Looking ahead now, the field's always changing. New tech like
Mm.
AI, nanotech, how does that impact this innovation safety balance?
That's the big question now. AI, for instance, has amazing potential. Identifying targets, predicting drug behavior, optimizing trials. It could really speed things up. Nanotech offers new delivery methods,
Oh,
but these also bring new safety questions. How do we evaluate the safety of AI-designed molecules? What are the long-term effects of nanoparticles in the body? Our evaluation methods need to evolve, and the regulatory frameworks need to adapt too, to ensure these powerful tools are used safely.
so the promise is huge, but we need safety science and regulation to keep pace. That balance remains critical.
Always. Innovation has to be tied to a constantly evolving understanding of safety.
So wrapping up this deep dive, it really comes back to that core tension. The push for innovation, for answers to devastating diseases, constantly balanced against the absolute need for safety and efficacy.
Yes, and we've seen how that plays out everywhere. Preclinical toxicity tests, ethical oversight in trials, regulatory gatekeeping, careful manufacturing, smart formulation, and newer approaches like QBD trying to build safety in from the start.
And it's crucial for you, listening, to remember this whole complex, often invisible process is directly linked to the medicines available and, well, to your health.
It is. It's a massive undertaking, driven by the goal of improving health, but always, always with safety as the anchor.
On that note, here's something to think about. As AI gets more integrated into discovering and developing drugs, how might that genuinely speed up innovation? But crucially, how do we make sure those vital safety checks aren't just maintained, but maybe even enhanced? What new ethical questions, what new regulatory hurdles might pop up as these powerful technologies become more central to bringing treatments to you? Definitely something to ponder, that intersection of cutting-edge tech and fundamental safety.